Hello everyone, welcome to my channel Cells and Genes. So today we learn about amino acids. We all know that amino acids, they are the building blocks of proteins. Building blocks of proteins. A lot of amino acids, they join together to form proteins. So, talking about amino acid, they are compounds that contains carbon, hydrogen, oxygen and nitrogen. So they are monomers of, they are also called as monomers of proteins. Let us see the general structure of a amino acid. So we have a carbon group here attached to its amino group, carboxylic group, the valence shell H, and here the R side chain. So this is the general structure of an amino acid, and this carbon is called as an alpha carbon. Okay. Now this R group is the side chain to which the other groups are attached, and coming to that, let us see general structure of an amino acid. This is the structure of lysine, and as we said that this is the carboxylic group attached, this is the amino group attached, and this is the alpha carbon. So these are the side chain, or this is the R group that we saw in the general structure. Okay, so this carbon is named as beta carbon, and this carbon is named as gamma, then sigma and epsilon. Okay, and here we can see that this carboxylic group is named as, okay, it is named as 1. Okay, so the carboxyl carbon as 1 because the C1, it is named to the carbon with the substituent contains the atom of highest atomic number. So those substituents which contains highest atomic number is given as a 1, so or it is given as C1, and here it is C2, C3, C4 and it goes on like that. So this is the structure of lysine. So here we saw how it is the general structure and how it is named. There are more than 300 types of amino acid in the biological system. Okay, so we know that there are 300 naturally occurring amino acids in the living organisms, but only 22 amino acid, they participate in protein synthesis. Okay, only 22 of them participate in the protein synthesis. And coming to the first isolated amino acid is asparagine. So asparagine was first isolated amino acid. It was first isolated from asparagus. Now coming to the classification of amino acid, we have two types of amino acids or amino acid classification. They are standard amino acids and the other one is non-standard amino acid. Okay. What is a standard amino acid? Standard amino acids are amino acids, they are incorporated ribosomically into proteins. So they participate in protein synthesis. Okay. So they are also known as proteinogenic amino acid. Okay. Now, what is non-standard amino acid? So they are naturally occurring amino acid and they are not incorporated ribosomically into proteins. Okay, so they are naturally occurring in a cell. So these amino acids, they occur naturally in the cell. Okay, they occur naturally in the cell, but not incorporated ribosomically into the proteins. But some are constituents of ribosomically synthesized, but some are their constituents of ribosomically synthesized proteins. Okay, some are constituents of ribosomically synthesized proteins. How they are a part of this ribosomically synthesized proteins? They are generated how? By post-translational modification. Okay, post-translational modification of standard amino acid. So they are naturally occurring inside the cell and not ribosomically synthesized or ribosomically incorporated into proteins. But some proteins are part of ribosomically synthesized proteins. How? They are generated by post-translational modification of standard amino acid. Some examples of non-standard amino acids is 4-hydroxyproline, 5-hydroxylysine, desmosine. Okay, then gamma carboxy glutamate. So we have some examples of non-standard amino acid. They are 4-hydroxyproline, 5-hydroxylysine, desmosine and gamma carboxy glutamate. Okay. So coming to the standard amino acids, we have 22 amino acids and here we see the structure of 20 amino acids. Okay. So the structure of 20 amino acids, as we say, they are being classified into certain groups. First one is non-polar aliphatic R group. So the example of non-polar aliphatic R group is, that is, they have the aliphatic side chains. First one is glycine, then alanine. So they have a CH3 group here. Glycine is the simplest amino acid. They have only the R side chain as hydrogen. Then coming to the alanine, they have a CH3 group as the side chain. Then it is a proline. Proline, they have a secondary amino acid structure. They have an imino group instead of an amino group. So seeing the structure of proline, we know that here we have alanine NH3 plus, but in proline we have NH2 plus. So that's why we say proline is having an imino group. Coming to the general structure, we have a carboxylic group and we have an amino group for all the amino acid except proline. Proline is having an imino group. Next, coming to the valine, we have CH, and C is branched to two other carbon, that is CH3 and CH3. And here lysine, this part is the R group. Then isoleucine, isoleucine is having a similar structure to leucine. Here is the branching CH3 and CH2 CH3. Then methionine. So all these are non-polar aliphatic R groups. So they have aliphatic side chain, so they are called non-polar aliphatic R groups. Next, coming to the aromatic R groups. So those amino acids with the aromatic side chain, they are called aromatic R groups. So those amino acids with aromatic R groups are phenylalanine, tyrosine and tryptophan. Now coming to polar uncharged groups are serine, this is serine, threonine, cysteine. So serine has an hydroxyl group here, threonine and cysteine. Cysteine is having a sulfidal group. Okay. Then coming to the other one is asparagine and glutamine. All these are polar uncharged amino acids. They have an uncharged side chain groups. Now coming to the charged, those amino acids having a charged side chain groups, they are positively charged side chain groups and negatively charged side chain groups. So this is a positively charged side chain group because it contains an amino group. Here also NH2 plus, and here also one NH is coming. So this is those amino acids with positive charged side chain are lysine, arginine and histidine. Now those amino acids with negatively charged R groups are aspartate and glutamate. So they have carboxylic group here. So they have a negatively charged R group, only aspartate and glutamate. So coming to the abbreviation or the one letter abbreviation, and we have three letter abbreviation. So the amino acids, they are mostly given in the abbreviated form. So in three letter abbreviation, it is very easy to understand. Only the most of the time the first three letters. Sometimes in the case of asparagine it is ASN, and in the aspartic acid case is ASP. So most, and here glutamine it comes GLN. So three letter abbreviations are most of the time very easy, but it is very important to understand the one letter abbreviation, because for alanine it is A, and for arginine it is R. Then asparagine N, aspartic acid it is D. Coming to glutamic acid, it becomes E. So it is D and E for the acid group. For methionine it's easy, it's M, and phenylalanine it is F. So it is also easy. Proline is P, serine is S, threonine is proline is P and serine is S, threonine is T. Tryptophan it becomes W, and tyrosine it becomes Y. Valine is V. Asparagine or aspartic acid, here it becomes B. So three acid, aspartic acid, glutamic acids D, E, and aspartic acid becomes B. Glutamine is acid or glutamic acid, it becomes Z or Z. So these are three letter abbreviations and one letter abbreviation. So coming to the 21st standard amino acids. So we now saw only 20 amino acids. 21st standard amino acid, they are 21st standard amino acid is selenocysteine. Okay, selenocysteine. cysteine. So we write it as SEC, or one letter abbreviation is U. Okay, and it has same structure of, or a similar structure of cysteine. The cysteine, instead of a sulfur, cysteine contains sulfur. Okay, but here the selenocysteine contains selenium. Okay, we can find the selenocysteine inside some inside the catalytic centers. Okay, inside the catalytic centers of some enzymes. Okay, so what are the enzymes that we can find selenocysteine as the selenocysteines? Okay, they are glutathione peroxidase. So inside glutathione peroxidase we can find selenocysteine. Okay, and next one we can find is formate dehydrogenase. Okay, so inside the catalytic centers of these two enzymes we can find selenocysteine. Okay, we can find selenocysteine inside the catalytic centers of glutathione peroxidase and formate dehydrogenase. So this is the structure of selenocysteine. We have the general structure of, this is the alpha carbon attached to carboxyl group and amino group, and this is the R group. So we have selenium. Okay, we have selenium here. So this is the structure of selenocysteine. Okay, now 22nd amino acid. Which is the 22nd amino acid? That is pyrrolysine. 22nd amino acid is pyrrolysine. So it is similar to, it is having similarity to lysine. Okay, it is having similarity to lysine, and it is present in bacterial proteins. Okay, it is present in bacterial proteins, and it is coded by UAG codons. So this is the structure of pyrrolysine. So we have the alpha carboxylic group, to which the carboxyl group and the amino group is attached, and this is the side chain, R group. Okay, so and this is structure of pyrrolysine, which has similarity to the structure of lysine, and it is found in some bacterial proteins. And this is the end of today's class. I hope you understood it. If you like my video, please hit the like button and do share my video, and also don't forget to subscribe my channel for future updates. Thank you.